Hi there, I am Saniya, and in this video, I'm going to be talking to you guys about how you guys can go from a SNP ID, single nucleotide polymorphism, like, one DNA base pair difference that's commonly found in at least 1% of the population around the world. How you can go from a single nucleotide polymorphism, either the rsID or the chromosome number and the location, how you can go from that information to figuring out the minor allele frequency for that SNP. So how can you figure out that information? And the minor allele frequency, guys, again, it's just like how often the second common allele for that SNP, how often is it found in the population? That's what the minor allele frequency is. The major one is the most commonly found in the world, and the minor allele is the second most common. And how often the second most common allele, so it can be either at a DNA base, it can be either A, T, G, or C. So either it's going to be one of the four bases is the major one, and the other one is going to be the minor one. And sometimes for some genetic information, you may not be always given that information on the minor allele frequency, but it may be super important for you to figure out, well, what is the minor allele frequency? So for instance, you might just be given, this is for Kunkel's Alzheimer's disease data set of genome-wide association study summary statistics. So you can retrieve this data set here from NIA, GDS. This is a really helpful site for IGAP rare variant summary statistics from Kunkel for Alzheimer's disease, for instance. So you might come across some summary statistics from the site here. And you can come across this data here, but on some p-values, and when you download it, it'll look something like this when you put it into a comma separated file, like it will open it in Excel. So you're going to notice that there's no minor allele frequency. Sometimes in some of the data sets, there are minor allele frequencies. So you may not necessarily need this information. It really depends on the luck of the draw. Sometimes you could be having a data set that doesn't have it, or sometimes you can have one that does have it. Like if we look at the let's see, if we look at the spinal cord, and this is some like EQTL data from GTech, for instance, and this is providing information for some of these SNPs. You can see it's not really properly aligned. Sorry about that. But what you'll see is that they can give minor allele frequency MAF. That's what they can report, this information. So, but sometimes some summary statistics for SNPs may not have that information on MAF. So you might be wondering, well, what is that? So that's what I was struggling on in my work, and I kind of want to share it with you because I think that after looking at different resources, I finally found one that is very helpful. So let's get started. And along the way, please let me know if you have any questions at all. And you can post a comment, and please let me know. So again, I'm just trying to share some helpful nuggets of information as I go through my journey. So you guys will also learn in this video basically how we can go from a microname to figuring out the minor allele frequency. That's one of the things we can go directly from the SNP IDs, or we can just go from there's another alternative that we could do, which is just going from chromosome and position information. We can go from here all the way to figuring out this information here, which is this. You can also figure out minor allele frequency as well. These are the two different alternatives. So I'm just going to show that to you guys. So let's get started. So what you will do is you're going to write virusmanager.install MAF DB GNOME 8B or the R2.1.gr38. So this is one of the new versions, GRCH38. So it's just going to take some time to download that on your computer using Bioconductor. If you don't have Bioconductor, please note that you can just use this installation. So this is a package that will help you get minor allele frequency data that from GNOM AD release 2.1 for this. So it stores minor allele frequency data from the genome aggregation database for the human genome version GRCH38. So it's really a very helpful package. So I made it on Bioconductor, of which I'm a huge fan. So we can just go through the PDF together and just see how that is. So the annotation package stores minor allele frequency data that comes from the whole genome variant set release 2.1 of the genome aggregation database. The data are exposed to the user in the form of a G-scores object named after the package and loaded into the main memory only as different chromosomes and populations are being queried. So, okay, so if you have a minor allele frequency that's larger than or equal to 0.1, you use 2 significant digits. Otherwise, if it's smaller than 0.1, we just use 1 significant digit to minimize space. So let's see how we can go through and do that. So there's like this nice little explanation here or example. So you can use whatever library of SNP looks, Homo sapiens DB SNP, rsCH38 as well. You can use this information here. Look up allele frequency for rs1129038, a SNP associated to brown and blue eye colors. So yeah, so some of these SNPs, they have these IDs, which are chromosome locations and positions, and where are the variations in the DNA. So that's how you can interpret the SNP. So we can also just look it up too and see. So we can see it's found in SNPedia as well. And it's part of a haplotype spanning 166 bases on chromosome 15, defined by 13 SNPs. Found in 97% of all Caucasians with blue eyes. So it's pretty strongly associated with the blue eye color. So yeah, we can just look this up, find the minor allele frequency here. Or if we have the information as well, we can also look that up too, because we are given that it's on chromosome 15, position 281111713. So we can do either or. We can have this coordinate here. So if we have SNPs by ID here, and we can look this up. So let's just try this out and see what we do get once we have finished running and installing. So I think it's just going to take a little bit more time. So in the meantime, I'm just going to just copy everything from there here, so we can just experiment with it and see how we can get those frequencies. So once we have this loaded, we can call library. Once we finish the installation process, we can just call the library package here. Hopefully I would have the SNPs locks database here, but if I don't have it, then I can always just run BioC Manager install, and then I can just call this inside here. If I don't have it, if I'm encountering an error, then I can just install it, and I can add this in quotes here. I just have to make this a comment. This is how you can install packages on Bioconductor. And having access to this kind of information can be really helpful, especially, for instance, if you're trying to do some like co-localization analysis, for instance, then you'd really need like the minor allele frequency if you don't already have that. So you would need this minor allele frequency. So that can be pretty important to have. And if you're a genome-wide association study or your EQTL does not have it, then you might need to just figure out for the SNPs, how do you find the minor allele frequency? So you might be wondering, okay, well, what is a minor allele frequency? So the way that we can just define it here is it's the frequency at which the second most common allele occurs in a given population. They may play a surprising role in heritability, since minor allele frequency variants, which occur only once, known as singletons, drive an enormous amount of selection. So if we look at like any genome-wide association study data, like let's say that we're looking at this set of Alzheimer's disease SNPs from from phase stage 1. This is kind of like how you may get some information. So I stands for insertions. So you can see that this insertion SNP in the effect allele, and then this is a non-effect allele as well. So the differences. So it's an insertion of ATC over here. This is a deletion. So that they're different things. But typically, if we just look at one base, the difference between effect and non-effect is one base. That's a typical SNP with an rsID. It's just at a chromosome in one particular location. Like chromosome 1 position, this position here will be having the SNP here. That's what it is. And then this is the beta standard error p-value. And it's sort of like a linear regression or some sort of regression that's comparing the individuals with Alzheimer's disease with the individuals who do not have it. And we're trying to determine, well, how impactful, how well can this SNP at this particular location and in this particular location in this chromosome, how well can it account for the differences that we observe between people with Alzheimer's and people without it. And that's what the p-value does. And this is beta that comes from a standard like regression equation. So I can prepare more videos for that too. But I just wanted to show you that, like, okay, well, what if we wanted to find the minor allele frequency? Which is what the MAF is. That's what we are trying to use this package for here. I think it's pretty much still downloading. So this is an extensive database, so we're going to have to just give it some time here. But luckily, it seems like the documentation is just these 3 pages here, so that should be really helpful for us. So it looks like we can just look it up, and it may take some time, but hopefully this will work out. So you can see that it takes quite a few minutes now, so you can notice the time difference. Maybe at least around like 10-ish minutes or so. We don't want to update them, so you can just click N for none. Okay, so now we finished this, so we can just comment this out. We can just call library here. And attaching this information here. Wait. I'll just also clear up my workspace too. So what we can just see is that, we have attached this library here. And then we also have library. There's no package called this. So there's no package here. So what we go, you see, if we don't have a package called SNP locks Homo sapiens, just human beings, this is what humans are called. So humans are called Homo sapiens, so the same thing as H sapiens, which is just humans. So this is the CH38, which is a more modern one. So we're just going to install it here right now, because we don't have it. So and then we can just comment this out so we don't keep reinstalling it. So we're just installing that, and just to kind of show you what is this exactly. So this is SNP locations and alleles for Homo sapiens extracted from this build 149. The source data used for this package were created by NCBI between November 8th and 16th, 2016. And the SNPs were mapped to a reference genome, which is this. Okay, that these SNPs can be injected in this or in this DS genome, the different data objects. So we're just going to wait for this to finish downloading. And you can use any SNP locations. I'm pretty sure that any will work, but that's just what they've given in their demo. And I just sort of want to, and I think it's important. You could probably even find like 149, 151. You could find different builds as well. But I think what would be very helpful is just to kind of replicate what they have over here. So yeah, let's just see how this goes. Hopefully it will work out, you know, fingers crossed. Let's just wait for this to finish downloading. So yeah, now we can kind of see here that, you know, the downloaded source packages, where are they, you know, like, so so I'm just going to click update all, none. Okay, great. So now I can load this in my library and it will work. Great. I can list, okay, this is this, okay, cool. So now let's try this out. Let's see, what is this? This is a formal class of D-squares. So it comes from broad in situ. This is a download URL. Download date is of October 12th, 2019. Some information as well on this data object. There's also a citation. So if you use it in papers, this is how you can cite it as well. Populations as well, if you want population information. Allele frequency, Africans, Americans, Eastern, Finland. I think there's a lot of different like locations. So we're just going to make a SNP database. So once, like, because this is like a database of information on different SNPs and it's a certain build. So we can just directly, like, kind of just to save some space. Instead, we could have had this in here, but just to save the space, they are having us just assign it into a shorter SNP database. That's what this is. So let's look at this SNP again, which is associated with that blue color in humans. Okay. So we're just gonna look this up, and remember these are kind of equivalent. This is the chromosome and the location on the chromosome. So again, that's the sort of so what this is this is the chromosome and position. That's how you can impute this 15 on this position on the chromosome. Okay, so now let's see, we get a warning message. So sometimes the heart sinks when you get a warning message like that. So let's just, you know, see what this is exactly. So it's always great, guys, to try out the examples with the tutorials. So here, unstitched G pause object with one position and two metadata columns here. Okay, so as we can see, what is confirmed here, that this is 15 and 28 11713. That's what this is. Yep, it matches here. Strand, reference SNP. Okay, alleles as ambiguous character. Okay. So now let's try the G scores. Wow. So assuming that GRCH37P7 and GRCH38 represent the same genome build between query ranges and the G score objects respectively, So once you look up the SNP of interest, you can then just query it here. So this is, you look up the SNP of interest and you get it into an object, and then you can just query that object here. So that's what you get here. And we get our allele frequency here, which we can use. Look up allele frequencies. And this will be the minor allele frequency here that we will be looking at, so 0.44. And equivalently, we can also do the same thing over here. For this G-range object, we get the same information here, excepting we don't have the reference SNP ID. So it seems that, SNPs by ID, but it looks up and retrieves this information, which is what this RNG object is trying to do, is trying to get the chromosome number, which is what SEQ names is, and the position is the position along the chromosome for this SNP. And this is the associated SNP that we had looked up. And what you can walk away with right now is this allele frequency. So this is the minor allele frequency. This is what we were looking at, what we were wanting. This is this minor allele frequency. This is the MAF, the MAF. Yes. So now, we just had this for one SNP. So if we want to have this for multiple SNPs, so again, these are identical, So if we wanted for multiple SNPs, instead of just having minor allele frequency for one SNP, then we can, for instance, let's say we're looking at, Kunkel's Alzheimer's disease stage 1 SNPs, guys. So let's say we're looking for this. Let's say we want to just select the first 4. Let's just ignore this indel over here for now. Let's just ignore that. So we're gonna just look at, because this is more like not a SNP, but let's just kind of focus on these SNPs here and try to get the information for these. Then let's just select these here. So that's what I did here. I just selected these and I just put them into a vector. So I got rid of this one and I just put these into this vector here below, called SNPs vector. So this is the same thing as this. And so again, guys, in this Kunkel site, what you will see is that there's no minor allele frequency information. So how will we get it? How do we get that information? Microname, SNP, ID, rsID as well. So again, this is usually rsID is a chromosome and a position. So we don't have the information for those. So let's just start with a really small example of just 4, and then you can extend this to as many SNPs as you'd like. So we're just going to read in the SNPs vector. So when we read it in, you'll see that it's over here. So SNPs vector is this. So if I call for SNPs vector, length of SNPs vector is 4, as we expect. Okay, great. So now I can call the same thing. Instead of SNPs for ID for one ID, we can call this vector directly, or we could just have had it inside here. We could have just pasted this inside here. Either or, this also works. You can see it might take a bit longer. So do expect that if you have a lot of these SNPs, it may take some time to run. So please factor that in. Or we could equivalently have, and we can also see what RNG is. It's extracting the sequence names, positions, strands, all that good stuff. So, we can see that is the case, or we can just also call this vector directly to save some space, and we're querying the SNP database here. Okay. And then you can just see these G scores here. This is this object here. So we can see the minor allele frequencies for these different SNPs here. So that's awesome. So where we previously didn't have that information for Kunkel, it's almost like we had this question, okay, well, what is the minor allele frequency? What is this MAF? And now, we technically could just fill out these 4 rows right now. This one will be 0.29. This one is going to be 0.29, yep, maps here, 0.29, and it's going to be in the order that we provided it, 0.29, 0.39, 0.10, and then 0.01. So then, that's the same process. We continue for all of these different SNPs here, but hopefully this will allow you to be able to just kind of extract the information on the minor allele frequencies that you need to do your analysis. So I really hope that this was helpful. This is again something that I'm discovering in my research, and I just wanted to share that with all of you guys, because I do think it's super important to be able to figure out how do you go from a SNP, either the location, like chromosome of the position, to the minor allele frequency, or the rsID, which is a SNP ID, to the minor allele frequency. It's super important for many different applications. Another thing I wanted to tell you guys, just because you might be, if you're like me, you might be a little, once you run this G-scores again and you have this, SNP information here, you might see that it's like a G-scores object. So let's assign this to just like some info. Right now, info is equal to this. Let's assign it to this, and we can call info again. I always believe you should assign it to a variable, so that way you can keep referencing it. You can write it out, save it as an object, as an iData object. So if we look at the structure, str, this is the data structure. We're gonna see, wow, looks kind of like an unstitched GPOS, and you might be like overwhelmed. So keeping it in this form, this object form, can have its advantages. But if you just want to have this information, allele frequency, just work with it very quickly, what you can just do is you can just say you can just call it like an info data frame. You can, all you'll have to do is do info data frame as data.frame info, and then you can just do like head info df or something, which will show you please see the first 5 rows of this info df. So you can just make it into a data frame and it will just work with you. So you can just see that it's like a data frame here. Now if you look at the structure of info df, it's something that maybe you can feel more at ease working with. So you can just see allele frequency. This is your minor allele frequency, and you can see this is 1, 2, 3, 4, 5, 6. So you can just go. You can also see call names of info df, and to make it more obvious, so this is the sixth one. You can just do number 6. You can just override it to being MAF, and then we can just look at the column names of this again. Just see the column names of this data frame, and I've changed it from AF to minor allele frequency. And now we can just view the head again, view the first 5 rows, and we can see that it's called MAF, minor allele frequency. So just trying to show you guys that you can totally convert it afterwards into data frame. Please don't be intimidated by it if you're not sure what the object is. You can just convert it to a data frame or a table or a data table, whatever you're able to work with, and then you can just continue working with that. And another very quick thing I wanted to show you is just some troubleshooting. If you are encountering a situation where you want to, like, maybe just you have a set of SNPs and you want to just find the SNPs that are beginning with an rsID. These are just a couple of very quick hacks. So if you just want to look for the SNPs that have it, you can just do the following. Let me just share that with you. You can just do the following, guys. All you can do is you can just like run this code directly. If you have a SNPs vector and you want to get rid of something that does not begin in that, for instance, all you could do is you can just we have a SNPs vector, and let's say that we did include Kunkles, that SNP in Kunkles that has this, let's say we included this, we can just like, and this was part of our SNP vector. Okay, so like let's say now that this is our new SNP vector, SNPs vector has this inside it, but we don't want to select it. We can just like call this function, which is just going to search for everything. What you notice, so sometimes when I use this grep function, it's like a regular expression, kind of a search kind of a function. It's like searching in this whole vector, and it's looking, this grep is basically looking inside this vector. You know, remember SNPs vector is all of this. It's looking inside and getting the locations of every single location in this vector, the index of every single location that contains an RS inside it. So if you were to stop for a second and just think about it, you would say, okay, well, what are some of the common features that that these ones that I want to keep? This one, like what are some, like I want to get rid of this, I want to get rid of this one above. And why? Why do I want to get rid of it? Well, it doesn't have an R and an S together. So I just want to look for the ones that have RS. I want to keep this, and this, and get rid of this one here. So I just want to only look for the ones that contain RS and keep those SNPs. So I'm saying this to you in case you have like hundreds or many of them and you just want to figure out the code as well, because otherwise you will probably have a mistake if you run it on this, because you might encounter something like this, where if you're running this code now with the SNPs spec, because the SNPs spec has something that does not have an RS in it, you're gonna get an error. So you want to instead now just get rid of that. You just only want to select the ones that have an RS inside them, and that's what this grep will tell you. It's going to tell you that in the SNPs vector 1, so remember the indexing starts at 1, which is a little bit different from Python, which starts at 0. So different languages, just a little bit to remember, keep straight between the two. So 1, 2, 3, and 5. That's why it said this. It did not have 4 inside it. So you can see it's just going to go through and just select you know, those ones, this one, this one, this one, and this one. So now we should instead query this, and we will have it work. And then you can just see your info, and all that stuff is great. The other thing I wanted to show you is that sometimes you may encounter this error if certain SNPs are not found. Just, you know, minor allele frequencies are not found for them. So they might ask you, you know, use info if not found equals drop. Just forget about them if you can't find them. So you just have to rerun this with this argument, if not found equals drop. But that's more of a judgment call. So that will be sort of based on what you're comfortable with. And the other thing I just wanted to show you is just like for your information, you don't have to be stuck with whatever we are looking at. You can definitely look at your own set of these. Databases. So for instance, the one that was being used right now was created. Okay, so I just kind of want to show you that you might want to keep on checking for the latest databases that are out there, because, for example, what we were using right now, which is 149, it may not be the most updated. So I just want to show that to you guys. This might not be the most updated. So it's 149 that uses this. So this one was actually, you know, the source data files came from 2016. So they are a bit outdated. So this is a build 149. And just as you would think, I think as this number 149, as this goes up, so you want to keep this, you know, you want to keep this straight whether you're doing. So one of the things that is important to know, guys, is that HG19 is the same thing as this. If you encounter that, The other one is just like, no, wait, I think, oh, sorry, I meant mainly like in GR37 is the same thing as HG19. So this one is more updated. So this one is equal to the 37 build. So that's what you want to incorporate. And so you'll see that like as this goes up, so you want to be cautious about this one, but in general, like, so you might want to keep this the same based on what your build is, like, you know, what data you're using, or you can do some leftover analysis there. But here, this is DB SNP 149. If you check out, just see if you're looking at like some of the recent ones, so you can check out online, see what there is out there. And you might say, oh, okay, wow, okay, this is 151. This may be more updated. And what we can see indeed is that this build is 2 years later, 2018 information. So it might be even better. So you might want to check this out instead. So if you want to use this updated one, then what you'd have to do is you'd have to just download it. Luckily, I was using that one. But I wanted to just show you what the tutorial was showing you as well. So all you would have to do is, if you wanted to use the 151, is you would have to just go here and then just do BioC Manager, install, and then write the 151 build, which is 2 years more recent, if you don't have it. But I already have this. So I'm just going to do library. So, oh wait, I thought I had it, my bad. So you will just install it, and then you can call the library, and then instead of calling this, you can just use this instead, 151, and you can just work with that. So you just want to keep on just to be really careful about what you're using, and I'm pretty sure it's going to work out for you. So just like be cognizant of some of these things that I learned the hard way. All right, thank you guys. So again, I am Saniya, and I'm very happy to help. And if you liked it, please subscribe to my channel. Please reach out if you have any questions at all. And I'm just really happy to help, and I hope that this can help you guys as well, who are visual learners and who need a little bit of extra help like me as well sometimes. So thank you guys.